Okay, thank you. So they're coming in, Dieter. I had to activate the webinar. So, so there's people coming in. Perfect. Perfect. So Stephanie, we're all set, I think. We can manage from now. Okay, so let's get started. So good afternoon. Welcome to this ERKNet webinar. My name is Francesco Emma. I'm speaking from my office in Rome. As always, before starting, I remind you that your microphones are turned off. During and after the webinar, you can send your question to the GoToWebinar application that appears on your screen, and I will ask your question at the end of the presentation. The webinar will last approximately 40 minutes, and we will have 10 to 15 minutes available for questions. So our speaker today is Professor Dieter Hilgers. Professor Hilgers has studied mathematics in Aachen. He finished his doctoral thesis at the statistical faculty of the University of Dortmund. Since 2001, he is the head of the Department of Medical Statistics at the medical faculty in Aachen University. He has been a coordinator of the IDEAL project funded by the EU, which has established new statistical methodologies for small population group trials. He's also the co-leader of the Work Package 20, which is entitled Accelerating the Validation, Use and Development of Innovative Methodology Tailored to Clinical Trials in Rare Diseases within the Horizon 2020 funded AJPRD project, and he's the co-leader of the Work Package in the European Rare Disease Research Coordination and Support Action Consortium. So from what you heard, Professor Hilgers is both an expert in statistics and in trials in rare disease. So he's perfectly suited for our talk today, which is entitled Overview of Biostatistical Approaches in Clinical Research in Rare Diseases. So Dieter, please.
So thank you very much, Francesco, for the kind words, and I'm happy to entertain you with statistics today. The title is already given, and let me go directly into the topics which I would like to cover. Well, I span the bow from having and analyzing natural history data to, let's say, learning about the content of clinical trials with respect to multiple endpoints and treatment allocation. Then it will come up to some design aspects. So 3 topics, and let us go directly into the depth. Well, if you have a registry data, which usually consists of longitudinal data, as you can see in my figure over there, so here in that one, you typically have courses of patients. Some you only see once, some you see, let's say, at several occasions. The time where you see the patients differs from patient to patient. So we are talking about heterogeneous time courses. And this also reflects in what you see on the figure over here, where we have from the ARCAS registry, a couple of 519 patients in, but let's have a number of 8 follow-up visits over 8 years. These are only 6. So how to analyze these data? How to deal with this heterogeneous follow-up times? How to deal with, let's say, if you have here a score like the SARA score, which has a limited range from 0 to 40, how you can deal with that. So to be a little bit more specific, what people usually do, yes, I have to mention that one. You see here that you see the first visit of this patient was at the age of 20 years. And this is visited first at the age of, let's say, 38 years. This is almost above the 50s. So how can you deal with that? We are talking about left censoring in that case, meaning that the first visit is completely different for all the group of patients. So a solution of that, which is very common to do, is that you choose a common zero time. So the first visit is taking as a zero time, and then the prolongation starts. And here you see the same data with a zero time conversion, and you see a lot of patients having only one visit. But don't forget about these patients. They are very important for the evaluation of that. So choosing a common zero time needs, let's say, some assumptions, and that means that the cause of the disease from that time point on is comparable. That means the age of onset does not play a role, but you can manage that by including covariables in your statistical analysis model. So another problem which you have with that one, which I depict just a couple of minutes before, was if you have a SARA score here, which is a very specific outcome measure for the in the ataxia field. It could be that a patient reaches the top of the SARA scores and does not change it. So here is a fictional data set. So one patient's scores here are the black points, and you see you have at certain times no SARA score present, and then the disease progresses and the patient gets worse, and then he stays at the highest score here, which is in that case a score between 0 and 4, which is a subscale of the SARA score. So if you apply the usual methods which you can do, you will get, let's say, a progression, let's say a linear regression function of this type, but you might be disappointed by the red curve. So you might feel that it would be better represented by the blue curve. We can do that, and that is typically termed as a so-called TOPIT model. And the TOPIT model is, let's say, a slight modification of what we usually do when you apply the regression model to the longitudinal data. It's just by a truncated distribution. So you truncate the distribution, the normal distribution, to the range which you observe. So even that, at the end, you can manage that. And in my next figure, I show you what we derived in the Friedrich ataxia field, together with my colleague Katrin Nitz here from Aachen, where we evaluated the EFACTS registry, 600 Friedrich ataxia patients, in the years 2016 and 2021 in the Lancet of Neurology. And then we built up a trial from these data. We developed the methodology to build up a comparative trial, which is the COFA trial. So to just explain the figure here, the total SARA score is depicted here over 5 years, and you see it is almost nicely behaves linear. So you have a linear progression in that. At least it could be interpreted as a linear trend. And you see the subscales of the SARA score, which is combined in that in these other categories. And we wonder whether the subscales behave as the same as the combined subscale. So what we found out when we used this methodology in our planning of a randomized clinical trials, we could apply that for making sample size justification. And it appears to be that if you increase the visit as well as prolong the trial from a 1-year trial to a 2-year trial, for example, it dramatically reduces the sample size. But you have to take into account that this behavior is very much related to the assumptions which you derive from your registry data. At that point, I would like to invite you to the first poll, and I have it here. But I think that Francesco can open the poll and you can answer the questions.
So do you want to read the poll? Would you like to read it?
Yeah, I can read the poll. It's basically on longitudinal natural history data. And the analysis of these data are meaningful because follow-up visits are always planned in time. Because observations below or above detection limits, field or float effects, do not occur, because information about planning and randomized clinical trial may be derived, or because considering participants' first visit at onset of the disease is always correct. Okay. I see the poll right now.
So
let's see the answers.
I'm waiting for, right now we have 41% of attendees that have voted, 47%. We usually wait 1 minute. We're up to 62%. I'm going to give you a few more seconds. See that it has stabilized. So I don't know what I've done, but I'll tell you this. Oh, sure, I don't know what I've done. I apologize for this. So let me tell you that 62%, so of the 62% that answered, basically more than 90% said that information about planning on RCT may be derived. That was the answer.
Great. That was the answer. Great. You got it. Well, I have to say there is another webinar which I gave within the EGP context, and there I give some more information. But if that does not fit to all your purposes, please get in touch with me. We just talked about the components of the SARA score. Well, in the neurological context, you have a lot of scores which have items, sub-items, and they will be summarized. So the main point, which I would like briefly to discuss with you, is a point with the multiple endpoints. Oops. Yes, multiple endpoints. Multiple endpoints are a big question, in particular in rare diseases. And there was a webinar from Johan Verbeek in the EGPRD context dealing with this point and statistical analysis aspect. A lot of guidelines concerning multiple endpoints are by the regulatory authorities like EMA and FDA. And we considered that problem, but unfortunately the terminology is not very, let's say, unique for all these points. So a lot of different names are on board, and even in the guidelines, and you never know what it appears and what they are meaning of. So we have the ISTOR paper in the ERKNet journal, which was recently published, where we worked together with Johan and Gert Molenberg and his groups from Hasselt and Leuven University, and we tried to make a uniform terminology and establish that in the rare disease context. So multiple primary endpoints, which we called, are those where you are interested in each component. That does not fit to the SARA score because we are not primarily interested in the items. It's less interesting. Then we are talking about co-primary endpoint. Oh yes, I have to say that multiple primary endpoints and the testing of each component is equally important, and usually they will be analyzed by Bonferroni correction for multiple testing. So co-primary endpoints are two endpoints which have equal importance, and they are tested simultaneously. And usually there is a decision rule connected to that, which means the all or none decision rule. And that means that, let's say the treatment effect is only decided if both endpoints reach a significant result. All or none procedure, no correction of the significance level is important. Another option is are ordered primary endpoints where you in advance establish an order of the importance of the endpoint, and here is so-called a hierarchical testing procedure to be applied, and even in that case, no adjustment of the significance level is necessary. Different other types are the composite endpoints. Composite is a multiple dimension endpoint which reduces by the occurrence of any of the components of the endpoint, usually not on the same scale. So a combination of, let's say, time to event data and continuous data. Multiple component endpoints are some sort of what you already learned about the SARA score. It's the components are combined within a subject to a single score or rating scale, and then the sum of this aggregated measure will be used to evaluate the data. We have different other forms of dealing with that. For example, the generalized pairwise comparison procedure, where Johan is an expert in, and also other procedures like multi-domain response index, which sums the scores of responses, are also sorts of endpoints which you can deal with. So all of these endpoints are important in rare disease field, and some of them apply also to so-called patient-centered outcome measures, PCOMs, or others. So again, we have a next poll, and the poll is about multiple endpoints. Multiple endpoint consideration in rare disease clinical trials are less important as endpoints are always simple, do not need specific attention in the analysis, can only be analyzed by Bonferroni procedure, or occur if PCOMs are considered.
We're up to 40% data of responses. I think this one is more difficult, apparently, from the previous one.
Okay.
Let's give us, so 54% have voted. We went up to 62% with the previous question. Let's see what happens now. As a matter of fact, the previous question, we had 83% that voted for answer number 3. Here I will be able to show you the results. So 60% have voted. I think we are more or less, it's more or less the same number than before. I'm just going to close the poll and I'm going to show you the results. Here we go.
Yeah, great. Great. Even that answer is very correct, because less important endpoints are very tricky to choose, and they need specific attention with the analysis, and the one-for-only procedure is not the solution anymore. So the last question is the right one. Thank you very much. Great. Great. Yeah. Okay. Let's continue. So when we are doing a randomized clinical trial, as I said, randomization is treatment allocation, and that is of particular interest in oops, I must go in the other direction, in that direction. And randomization, well, what we found out in our research has a special attention to be taken in the context of rare disease field. So, well, what you see is, or what we all know is that randomization should avoid or limit or mitigate bias in clinical trials. So that's the story. And the problem is whether it can do. And bias is some sort of distortion of the primary endpoint variable. So the impact on the primary endpoint variable, or the impact on the treatment difference, that is the important thing which should be studied. And basically, nevertheless, randomization has something to do with the statistical inference, but that is not my primary interest right now. So what we found out in our research is that depending on the sample size, we have here a study of 8 subjects, here a study of 20 subjects, here 40, here 80, here 200. You see here the effect, the impact of the randomization procedure on the type 1 error probability. It should be 0.05, here the red line. And we do some simulation study where we pose some bias in that. And what you clearly see is that in smaller trials, the bias has much more variability. So the effect is much more unpredictable in advance. It stabilizes when the size of the study increases. And then that brings us to the statement that one has to pay attention to the selection of randomization procedure, in particular in small population clinical trials or in small trials. Be aware, this is permuted block randomization, and there are other randomization procedures available in the literature as well as software. So what we, from this figure in the background, developed is a procedure which is called the ARDO procedure. It's the evaluation of randomization to design optimization. And we use this as part immediately following the sample size justification in the clinical trial development process, the randomized to study the effect of the randomization procedures or different randomization procedures on the trial results in the simulation study. The same approach can be used to derive, let's say, a bias-free estimate at the end of the trial. So both things can be done with this procedure. And we have a nice paper about that where we show our methodology, which you can follow. Clearly said, this depends on the trial design, this depends on the endpoint, this depends on the number of observations, and at least on the effect you will study. And we investigated already some situations, a 2-arm single-center clinical trial with continuous endpoint, a 2-arm multi-center trial with continuous endpoint, multi-arm single-center trial, and 2-arm single-center trial with time-to-event endpoint. And what we do recently is we considered also multiple endpoints with a two-arm parallel group design. My co-worker, Daniel Bodden, is working on the group sequential design, which can be extended to platform trials. Here is some work which I will do in the next time: crossover trials, allocation ratio, dependency on that, two-arm trials with, let's say, platform trials with response adaptive randomization, linear mixed effects model. So a lot of things to do there, and there's also a webinar specific to that point in the future. And with that, I come up to my third and last poll, and then I will go to the design case. But before that, let me briefly ask you the poll number 3, the randomization procedure for treatment allocation in comparative trials should be selected by the availability of software, need specific evaluation in rare disease trials, is limited to permuted block randomization, mitigates bias always.
So 40%, Dieter, right now.
Great.
When we reach 60%, I will close the poll.
Great.
Okay, I think we can close the poll. We're at 59%. Okay, perfect.
Great.
Here it is. I'll show you the results. Can you see them?
Yes. Okay. Well, again, the majority has voted for the correct solution, needs specific evaluation in RD trials because you see the effect. Some people do that commonly decide about randomization by availability of the software, but that should not be a reason for that. Permuted block randomization is the most known procedure, but it is not, the spectrum of randomization procedures is not limited to that. And again, I showed you that the ability or property to mitigate bias, it depends on the very specific clinical situation you have. So with that, I would like to continue the discussion and go to the confirmatory trials. And I would briefly just remind you that we in statistics do have 3 potential specific hypotheses we can prove. Usually, everybody is interested in a superiority trial where you prove the superiority of one treatment compared to the other. But there are also some trials which are dedicated to showing the equivalence of two treatments or the non-inferiority. So as these terms, you see in yellow the yes and no answers to the questionnaire, which was submitted to the lecture in January. Most of you are familiar with that. I will come to, let's say, specific design questions. I will start with the most minimal design you can have, the N of 1 trial. Then I will go to the crossover trial and end up with a parallel group design where you borrow information from historical control. So what is it? So N of 1 trials, which are now termed, go back to a paper of Guyat in 1986 in the New England Journal of Medicine. And here it turns out that it is determining the optimal therapy within an individual patient. So randomized trial within an individual patient. So N of 1, usually the first started with N is equal to 1, but because of the equal sign, which does not fit to a title in a journal article, it was substituted by N of 1. So you only have one patient in the trial, but you have the ability to change treatments after a certain amount. So N of 1 trials are very important right now and sometimes discussed. And if you talk about that with a statistician, we probably might have completely different things in mind as what is turned out in the literature right now. In the ideal project, Stephen Sen did some work in that perspective, and he can be called as an expert in that perspective for a long time. And we have also a webinar of Patrick Ogena in 2023 about N of 1 trials in the psychological area. N of 1 trials are typically multiple crossover trials, usually having randomization, so randomized treatment allocation, need blinding, and are conducted in a single patient. Sometimes an N of 1 trial will be conducted in parallel in several, let's say, centers and then aggregated together. But that is another extension of the design. So major design elements are balanced sequence assignment, blinding, randomization, and systematic outcome measures. And I show you a picture of that, and you always have to keep in mind that the aim of this design and the conclusion from that is only to find the optimal therapy within that single patient. Okay, here you see the sequence of two treatments, A and B. So we have A, B, B, A, B, A, B, which are given in a random order. Usually, this is called, we do some cycles. So two treatment periods are combined to a cycle, and then we compare the treatments within the cycles. So that is a very typical picture how the design works and how one can compare. Typically only two treatments, and that's nice. So N1 trials can be considered as a special case of crossover trials. Crossover trials do something similar. They do, let's say, changing the treatment within a particular patient at particular periods. So to show you what I mean with that is, let's consider here the simple crossover trial. Here we have the simple crossover design is a standard 2-period, 2 sequences, 2 treatment design. And that means I have one treatment period and then potentially with a washout period, a second treatment period later on. I must make sure some conditions. And we have one sequence or one group of patients who are randomized to the sequence AB, and the other one is randomized to the sequence BA. So 2 treatments, AB, 2 sequences, 1 and 2, and 2 periods, and that is the characterization. So what do we have to have with this crossover trial? We have a stable or chronic disease, because the patient must have the same disease status at the beginning of period 1 than he has in period 2, and vice versa. Because otherwise you get a shift in the response independent of the treatment allocation. So no carryover, no residual effect of the effect of period 1 in period 2 should occur. We call that carryover effect. And no total curation of the treatment. These are conditions. So some people argue that in rare diseases, this is the best design. But don't forget about the assumptions you have to make. So this simple design has some drawbacks because if you have a carryover effect, you cannot estimate the treatment effect in a proper way. So an extension might be a 3-period design. So a 3-period design is one design which consists of 3 periods. If you have only 2 treatments, then a potential solution to allocation of treatment to patients could be you start with A, B, B, and then in the second sequence you have B, A, A. It turns out that the design which you might have directly in mind, which you think which is more better, is the design A-B-A versus B-A-B, but it is worse considering statistical properties with respect to the power of the trial. So that means you must need much more patients. So the positive point in these designs is that the patients act as his own control. So confounding is mitigated, variance is reduced, heterogeneity is ruled out, and sample size is reduced. Well, when I'm talking about sample size, what does that mean in numbers? Here you have a comparison of several designs which I elaborated for you. Please take into account you have a trial with 80% power and 5% significance level, and you have, let's say, medium effect size of 0.5. So very specific examples, the assumptions, and you will do the analysis by, let's say, some sort of t-test. Very specific t-test for the crossover. So in a parallel group design, you need 128 patients, in total of patients in your group. In a Bennet scenario, 64 in each group. You see that it doesn't matter whether you include the baseline values. Baseline values only make your trial much more expensive because you have to take into account the baseline measurement on the cost characteristic. So now I'm going to the simple crossover, ABBA, the first red one here. So you see that the same treatment effect I can establish with only a quarter of the total sample size of the parallel group design. And that means I have 16 in each group. So that reduces the total sample size dramatically. Also, the measurements you have to take into account are 64. So you can further improve. By the ABB-BAA design, you only have 24 observations in total, 12 per groups. That even reduces the number of observations. Whenever you can do that and you don't have so many dropouts. So the number of observations here is a little bit higher than in the ABBA design, but it has the advantage that you also can, let's say, estimate the carryover. You can do some further improvements. I call that the Cologne against the Swedish pop group. So we have the Swedish pop group, it's ABBA, and the Cologne pop group, it's BAB. So this is my design, ABBA versus BAAB. So again, we can reduce the total sample size by, let's say, to 16 patients. It's half of the ABBA design. That means 8 per treatment group. Whenever you can do a trial like that, but you have to take into account that the number of observations you have to measure at each individual does not decrease so much. It's 64 still. So that means go to a well-trained statistician and discuss with him whether this design could be applicable in your case. And you even have to take into account that sometimes endpoints are trained. So, for example, Jörg Schulz told me once that if you measure the SARA score every 3 months, every patient would be trained with that endpoint and will improve irrespective of any given treatment. So with that, I will come to my last point, which I would like to bring to your mind, and that is a very special group. If you would like to do a parallel group design, sometimes you have, let's say, my initial longitudinal natural history course data. And you might think, okay, I can put the natural history course data and borrow this information into my randomized. Well, that's possible right now. The FDA has a guideline in 2019 about rare diseases, natural history studies for drug development. And they recommended to do a hybrid approach, of using external control data to add to a concurrent randomized control arm in a clinical trial. And that might be a useful approach. Before I come to that, explain to what that does mean, I will briefly refer, there are plenty of potential analysis procedures you can do. Let's say Bayesian method with power prior, meta-analytic, predictive prior methods, and as well as frequentist methods like the method from Thiele in 2024, which means pool then test approach. Pocox in 1976 has formulated criteria which must be fulfilled for the control data so that it is meaningful to pool them with the randomized control data, because that is a very tricky thing, because patients might differ from characteristics and so on. So what is our approach? Well, our approach is as follows. And this is work from Steffi Beat, which is my co-worker, and we worked that together with Martin Posch from the Vienna University. So we are first thinking about the superiority trial of an experimental versus a control group. And we think and try to do the approach that will reduce the randomized clinical trial sample size so stop the trial at a certain extent and show whether we can pool historical controls which we have at hand and selected before the trial starts, and do the analysis with that. So that means we start the trial, we plan the NA, the sample size of the final evaluation. We make a stop at a particular point in time. Then we try to figure out whether our controls of the randomized part and in the historical part are similar. So that is an equivalence test, as I mentioned just before. If the trial says yes, they are equivalent, then we do the final analysis. The NE patients, let's say 50 patients against 650 controls. This is, the numbers should be selected that the power requirement is fulfilled. Then we do the pool then test approach. Otherwise, if the equivalence test say no, not equivalent, then we fill the control and the experimental arms until the final sample size. So it turns out that this approach can be done, but we are not so, let's say, enthusiastic to say, okay, you can do that in every case, because there are some drawbacks in that which needs to be considered. So one of the potential drawbacks I would like to bring to your mind is that, here are our numbers. We would like to do again a trial where we would like to establish an effect size of 0.5. And in a parallel group design, we found that 38 patients must be included in the trial with a power of 90%, reaching a power of 90%. So our decision rule is that we have to look to the data at midterm, at 74 patients in total, which is roughly a little bit more than half of the patients we needed to recruit in total, and then do the let's say equivalence test. So what we then have is we have a trial with 74 patients in total plus the 500, so 574 patients when the equivalence test says yes. And this has also, for the final comparison, a power of 90%. So you see that this design is dramatically imbalanced, which does not matter anything because the power requirement is so good. You have less information for the new treatment, but you can stop your trial at that point. So what we think is the hybrid approach in a frequentist setting is possible. We have to take into account that the last test proves a non-inferiority hypothesis and not a superiority hypothesis, which might be a small drawback. We found out in our simulation study that at least 50% have to be recruited of the 50% of the final or initial total sample size has to be recruited until the first evaluation. And there might be some slight power. So we think that in situation where you have, let's say, when you have, let's say, the option to include control patients, that might be an option if you don't be able to do the total sample size in a reasonable time. With that, I put my summary slides, and I'm open for questions. Linear mixed effects model, as I introduced just in the beginning, are a suitable way to analyze longitudinal natural history data. The ERKNet approach is a fine approach for selecting best randomization procedure and improve the characteristic of your trial. There are several ways to analyze data from multiple endpoints, and that could be that should carefully be decided on. And then we have N-of-1 trials, which is which is the optimization of treatment response in an individual patient. The extension of that are crossover designs, and crossover designs can be improved by extending the periods to a certain extent, and historical controls could also be possible for the use of efficiency. But the value should not be overestimated. With that, I thank you for attention, and I also thank you for answering the poll, and I'm open for your questions. Thank you very much.
Well, let me show up my screen again. Do you have a last slide after this one? Often we have.
Yes. Oh yes. Yeah. We have to. You know, the next webinars are the 18th of June from renal tubular. Oh, that's difficult for me. It's from and then a talk on the 2nd of July from Karl-Peter Schlingmann from Münster, and the 10th of September of MGRS, that is easy for me, from Roberta Fenoglio from Turin.
Thank you so much.
Here are, let's say, the QR codes where you can follow the webinars here. Sorry about that. Thanks for joining.
Well, this was really enlightening. I mean, I really enjoy your webinar. Thank you so much, Dieter. We have 5 minutes for questions, so I don't see any coming. But if you have some please answer them. I do have some questions for you. So one question, in rare diseases, the problem is the sample size. I mean, it's clear this is a problem, and then sometimes we're confronted into, making some pilot studies to see if there is a signal there. And so what we usually do is that we sacrifice power and we say, look, let's treat 5 or 6 patients and let's see if we see something. And then, and there's 2 things we can do. And I was just wondering to see how statisticians like you would react. So one of the things is that we can say, okay, let's see that usually these are severe disease, by the way, so you really want to make a difference. So sometimes we increase the effect size. We say, look, if we need to have a treatment for this, it's going to be so difficult, at least it has to have a huge effect. And so this is how we try to power our study. We say, look, we want an effect size of 1, 1.5. It's just go. It has to be really significant. The other thing is that we do pilot study, and I think that in that often we make a mistake, in the sense that we say, let's treat 5 patients and then see how it goes. But then if you want to continue or to expand or to make another trial where you use these patients to extend your number, you need to have you need to think about your protocol in that perspective, that trial could be extended. So I was wondering to have your input and your on the effect size and on the design of conditional trials where you just do a step and then according to the result you decide to continue. So I just wanted to have your comments on this.
Yeah, thank you very much, Francesco, for your question. Well, in modern statistics, we try to put this in a context of what we call adaptive designs. So meaning that you are allowed to do a pilot study, but the whole project will be formulated in advance. So you say, okay, I will do a pilot study, and based on the results of the pilot study, I will continue or stop the trial, or I will go in another direction. For example, what the people, my academic teacher, Peter Bauer, was a former of this sort of trials, and he said, well, you do a small trial, and if you see an effect, you can stop. Don't. If you not see an effect, if you see, let's say, a trend in, you should think about a second trial, which, let's say, also can have a different endpoint, also can have a different, let's say, design, to go from a parallel group design or crossover design to a parallel group design. The only thing he puts is combine the p-values of the of both sub-trials and combine them to fulfill at the end the 5%. You even can have the decision after the first trial to stop for futility. So this can be done, and this can also be done in a broader context. Nowadays we call that master protocol or platform trials, where you start with a certain amount of different treatments. You might have heard that in a COVID pandemic, where you start with 5 or 6 treatments and then continue, then you drop out some of the treatments because they are not successful. You have new treatments coming into play. So that if you have a plan of that in advance, then that can be used. But it needs some further and thorough, sophisticated thinking about what could happen in the trial. But yeah, possible. Okay. Not easy.
And then maybe one last question that was not clear to me. it's about the carryover effect in a in a crossover trial. So when you do a more complex design, like a you know, an ABB versus BAA design, do you control for the carryover effect by doing this, or do you not?
Yes. The problem for the statistician is if the effect cannot be separated from each other. We mean if we estimate the treatment effect in the ABBA design, then it is always biased by the carryover effect. This design cannot separately estimate the treatment effect and the carryover effect, and so it cannot be distinguished from each other. But extending by one period, you can do that. And that's fantastic.
Okay, okay. Well, wonderful. Thank you. I was actually really enlightened, and I really love to hear your talk. I see that there are no questions, but that people have not left the webinar, which means that they were still interested in listening to your answers. And I think we have come to the end of the time that was allowed to us. Thank you again so much. This was, I think, a wonderful webinar, and I think we as doctors really need to be more prepared in understanding your language, so that we can answer, we can ask you the right questions. I don't think we will ever be able to replace a statistician. I think you are absolutely needed, but it's also very important that we understand your language, and these webinars are very useful. Thank you very much, Dieter. And thank you all. Okay.
You're welcome.
Bye.
Bye-bye.
Bye.